Stanford University.
Both the U.S. and the Japanese send out expeditions to Antarctica to look for meteorites. And the way they find them, they've learned a long time ago that there are these blue ice fields where the ice is actually blue. And it is in these blue fields that rocks that were trapped in the ice by whatever means are brought up to the surface, and then the wind and sublimation remove some of the ice, leaving these rocks behind. So every year, if you go to a blue ice field, even the one that you went to last year, you will find new rocks. And the way you find them is just look around, and any black object, you can see these sometimes from up to a kilometer away. Any black object is likely a rock, and most of them are meteorites. There are some rocks that come from mountains and so forth, but each year the U.S. brings back hundreds of these, typically close to 1,000, and the Japanese the same way. Well, a very small percentage of those are Mars meteorites, usually well under 1%, but they're enough to make it worthwhile, and there are a lot of people that study the other kinds of meteorites. And the meteorites all come back to Houston, where they're roughly described, and many of them are chipped a little bit so you can make thin sections and see what they are. And then most of them are sent to the Smithsonian, where all U.S. meteorites officially belong to the Smithsonian. The Mars ones we tend to keep in Houston because we have these clean cabinets and we're set up to process them. The Japanese do a similar thing, and they have their expeditions are sponsored by the Polar Research Institute of Polar Research, and they bring back the meteorites and keep them in cabinets there in the Polar Institute. Okay, there we go. The point I want to make with this slide is that we have a nice team that has been working this problem for now about 12 years, and it's not just me, it's a whole team of people, and including even a company is helping us. In '96, there was a big press announcement that some of you may even remember, in which we announced our findings in Washington. And indeed, that was a terrible circus with hundreds of cameras and lots of questions, and the head of NASA and a few skeptics like Bill Shopp, and I'm not sure we ever recovered from that. It was a big news event, and it was clearly something that got into the media in a big way, and that was not really our intent, but that's what happened. And the point of this is that the press blew it up much more than what we had actually said in the paper. If you read the paper, it was fairly conservative, and the press is not used to gray. They like black and white areas, so that's what happened. And we got accused of pumping up the NASA budget, which is what this shows. And indeed, although that was not our intent, that's what happened. The NASA budget got a pretty good infusion because of this paper and all the publicity with it. And in fact, the whole Mars program got an infusion of money that has continued to this day. And the whole Astrobiology Institute was started as a result of this paper. And so this course can trace probably a lineage to this paper. Right or wrong, it put a big bump in the science of life on Mars. All right, on to the technical aspects of it. We really had 4 arguments in that paper, which are given here. There were these carbonates, and we argued they were formed at low temperatures, possibly with the assistance of microbes. There were possible microfossils, what we now call biomorphs, things that look like remain like some trace of life. There were polyaromatic hydrocarbons, or PAHs, and we found those with the lab of Dick Zare here at Stanford, and we found that they were closely associated with those carbonates. I'll say a bit more about that later. And then we found these little tiny magnetite grains, nanophase magnetites, and it was noticed that by our team that they were very similar to magnetites known to be formed by a certain kind of bacteria, magnetotactic bacteria, which had been known for about 20 years before our paper, that there are bacteria that produce in their bodies these little magnetites, which are used like compasses and are used to navigate and find the optimum environment that those bacteria thrive in, which happens to be where the reducing, oxidizing chemistry is such that they can make a living off those oxidation reduction reactions. And we argued that this suite of features taken together provided possible evidence for biology on Mars, ancient life on Mars. Now, we also said that there was no single feature that was definitive for biology, or conversely, we argued then and do today that if some of these features can be formed by non-biologic methods, that does not invalidate our hypothesis. The hypothesis will still stand unless you can prove that all these features did form by non-biological processes. And we had a lot of supporters in the science community and a lot of detractors who the main argument against it, as we'll see, has been by a series of research groups that have argued that the magnetites can be formed by non-biologic methods. Now, if you think about what I just said, does that invalidate our hypothesis even if true? And the answer is no. It's just an alternate explanation for the magnetites. But it does weaken our argument that these features are supportive of biology. So we'll get into that a little bit more. There were some very early objections to our hypothesis. The meteorite was probably contaminated in Antarctica. This was found in Antarctica, and maybe the carbonates precipitated and grew there, and maybe the polycyclic aromatic hydrocarbons were really dissolved in the meltwater and precipitated out on the carbonates, and that's why they're there. And maybe the magnetites were from either wind-blown dust or from yet undiscovered magnetotactic bacteria that lived in Antarctica and got into the meteorite from the environment. Another argument against us was that the magnetotactic bacteria that we didn't specifically say this, but since Mars, at the time we wrote that paper, was thought to have no magnetic field at all, why would magnetotactic bacteria bother to develop on Mars by evolution? There's no forcing environment there. Well, 2 years after we published the paper, in fact, some very strong magnetic regions were found embedded in the rocks on Mars, and the magnetic experts pushed those back to say that meant that Mars had to have a strong dynamo, just as the Earth does, only probably stronger in the early days. And that dynamo stopped at some time, but it left its imprint in the rocks. So in a way, the presence of magnetotactic bacteria, magnetite, if that's what they are, was a clue that Mars had a magnetic field at one point, although we certainly didn't know it at the time we wrote the paper. We were also criticized initially by those that thought these carbonate globules formed at high temperatures. There are, in fact, a number of carbonates that are formed in volcanoes, for example, by igneous processes. We were criticized that the microfossils were coating artifacts and not real, and I'll get into that. The microfossils were too tiny to be real. That was another big criticism. And so we had our hands full at first looking at all these criticism areas and either trying to answer them or conceding that, yeah, that's right. All right, I want to talk briefly about the temperature formation here. Several authors came out within a short time, 96 and 97, and proposed that the carbonates formed at high temperatures. And if true, that would weaken our hypothesis. Wouldn't invalidate it, but would certainly weaken it. And these papers were given very wide play in the media and at some scientific meetings. And it started something that we've been fighting ever since. It started the development of a consensus that we had been discredited because of the huge amount of publicity our critics got in answering our hypothesis. Now, I'm not saying that's wrong, because we got a huge amount of publicity when we first came out with the hypothesis, but there were many people that also got publicity because they seemed to contradict what we had said. But it turned out that within a few years, the pendulum turned the other way toward low temperature origin, and in fact, one group did a complete 180 and came out with although they had argued for high temperature, they wrote a paper a couple years later that said, oh, low temperature is what it really was. So, but the problem is that these reversals in the science didn't make the media, and to some extent didn't make the scientific meetings with the splash that the original papers had. So even though we were supported by all the later data, we were still tarred with the earlier papers, which turned out to be not correct. This is just some of the reason why the temperatures were never very hot when the carbonates were formed. The oxygen isotopes, once they were measured in detail, showed that they could not have been hot, the carbonates could not have been hot, or the isotopes would have entirely different patterns. The polycyclic aromatic hydrocarbons will not take high temperatures. They degrade and disappear. And the people that studied them said that based on the structure of those PAWs, the carbonates were never above a couple hundred degrees. And if, as I will get to in a moment, if the magnetites were formed by cooking the carbonates at high temperatures, the magnetites would be quite different from the ones that we saw, and I'll talk about that. So anyway, low temperature seems to be accepted. A lot of this story is in this book. I don't mean to plug this book necessarily, but some of you have seen it, and it's a good example of how science works and how science and the media and the politics all intertwine. And if you want to see a case history, this is a good thing to look at. It's available from, I guess, from Amazon. The other thing that's interesting is there's a PhD thesis published now based on this interaction of science and politics and the media. And I can give you that reference later, but it's a very interesting thesis, and it concentrates mainly on the political aspects of it, and also the politics of the big journals like Science and Nature, and how they react to premature release, and how the pressure was on from NASA for premature release of the data, and it's a very interesting story.
Do you feel comfortable just saying briefly how that all happened with the premature release?
The White House released the story because of a guy named Dick Morris, who was an advisor to the president, and who had apparently whispered it to his female companion, who called the tabloids and revealed it, and got it all wrong, but it came out. And when that story started to come out, the CBS and the networks started talking about it, and at some point NASA said that we don't want this to dribble out in the tabloids. We have to have our press conference and release it properly. And I was on vacation and on camping on a river in central Texas, and I was contacted to get my behind to Washington for this press conference, and to heck with my vacation. And so that's what happened next. And science was not happy with it, the journal was not happy with it, but they accepted it. And after negotiation between science, the journal, and the NASA public affairs and top management. So it was all very, I think most of the story is in that book, in fact. All right, so let's move on to the magnetites. Because I want to point out, and I'll be happy to discuss some of these other features with you about the size of the fossils, microfossils, whether they're really microfossils. I'll be happy to discuss the other contamination issues with you. We essentially wrote papers that disproved all those contamination issues, and those are all published, and in fact nobody disputes that anymore. Nobody argues that any of this stuff was formed in Antarctica anymore, although those were big arguments at the time. The only controversy that is really left here, and aside from the morphology of the possible microfossils, which I can get into in a minute, but the main controversy has been the origin of the magnetites. And we argued that these magnetites were formed on Mars. Nobody takes exception to that now. And furthermore, that they were detrital. That means, in geologic terms, it means that they were washed into the carbonate and were deposited there in cracks and crevices. And there may have been some of these bacteria, magnetotactic bacteria, in there, but there were also a lot of magnetites which are not necessarily associated with the magnetotactic bacteria. And so how do we know all that, and what is the evidence for that? Okay. All right, this is just a picture of the rock itself and some of the magnetite carbonate globules. We call them pancakes now because we finally realized they're mostly rather flat, like pancakes. They're not globular. And we can show, I think, and I'll show a few pictures to support this, that there's a lot going on. These carbonates are some of the most complex carbonates that have ever been studied, and these have been studied by more than 100 research groups throughout the world.
Dave, I'm sorry, can you go back to the picture before? cores. I went through that in class a couple of weeks ago, but could you just explain what they're seeing on the right again?
Okay, on the right, those little gold parts are the cores of the carbonates. Those are mainly calcium carbonate with some magnesium and some manganese. The dark bands are the iron-rich bands, which are also mainly carbonate, but are full of these magnetites. The white band is a magnesium carbonate, which has no color in it, then is fairly pure. And then the outer dark band is another magnetite-rich layer. Now, there's also some zoning in the interior of the cores, but for example, the very center is usually enriched in manganese. Now, we're still trying to understand the details of all this zoning. Well, we understand how it goes, but we're trying to interpret it. It's hard to interpret in detail. Okay, there are a number of features here. I've already talked about the zoning. I'll show you the chemistry. This is a chemistry of one of these features, showing for example, the manganese-rich layer in the interior on the left, showing the iron-rich layers, and so forth. This is a standard electron microprobe dot map, which is not real clear because of the black background. The one without a label on the bottom is sulfur. This is a drawing or a cartoon of how these things really are. The one on the left is an actual SEM picture with the carbonate artificially colored in. The one on the right is what our critics have been able to produce in the laboratory. And the difference is that ours, the real Allen Hills one, is always embedded in a pit, a form-fitting pit. And that does not occur in the laboratory versions. In the laboratory versions, the carbonate forms in cracks between two pyroxene surfaces. And why is that? That's fairly critical. And our hypothesis is that pit is made by the microorganisms, and it's made by acidic dissolution enhanced by biology, so that pit is actually dissolved into the pyroxene by the microbes. That's our hypothesis. And they're always like that. We've never seen them like the ones at the right. And that can be seen in some of these detailed textures. These are all carbonate surfaces. And all the carbonates in Allen Hills show some water dissolution. The carbonates are where two parts of the meteorite have pulled apart, and it leaves these little carbonate discs somewhat embedded in the surface. And in every case, the surface of those carbonates has been somewhat weathered or dissolved by water. This shows a very abrupt region between the iron-rich carbonate on the right and the pyroxene or silicate on the left, to show that this is where the carbonate is sunken into the pyroxene, and this is what you get if you look at the top of that pit. The pit is filled with carbonate, and the pyroxene on the left is not particularly modified, but on the right you'll see that the carbonate is there and it's been modified. I'll get back to some of that in a moment here. This is another part of it. This is the rim between the magnesium carbonate on the left and the iron-rich outer rim. In other words, between the white part of the Oreo cookie and the black part of the Oreo cookie. It's a very abrupt boundary on the scale of essentially tens of nanometers. It is not a gradational boundary. It is extremely sharp as these things go. And that gets into our argument later that these things have not been heated. They have not been heated after formation, or that boundary would have blurred together by diffusion of the elements because of the heating. The point being that this is an extremely sharp boundary, and that tells us something. This is simply the texture of the pyroxene on the left, away from the carbonate, and on the right, the carbonate is colored brown, but what you see on the right is underneath the carbonate, where part of the carbonate is broken away. You can see it's a different texture. It's different between underneath the carbonate and the pyroxene on the left. What's that mean? Well, it means we think that there's been a different process working on the right-hand side of the screen. There's been possibly organic acid there, which was not on the left. And that remains to be shown, but that is our hypothesis for that difference. This is a closer view. On the top is the surface where the carbonate's been broken away. And you can see it's got a very complex texture. And we think that texture is a result of the acidic dissolution caused by the acid associated with the carbonate. And on the left is the carbonate itself, and you can see how the two fit together. We have actually done a detailed study of these carbonates with the TEM using the focused ion beam or the FIB technique. We've cut little slices of them here and there and looked at the slices. And this is a cross-section of one of those slices. And there are several interesting things. The iron-rich area has been colored in brown. The uncolored part is the magnesium carbonate with magnesite, and it is those brown parts that contain the magnetite. And we're trying to figure out how that works. This shows where the slices are made. And this is the detail, again, of one of those slices. And what can we learn from this? All right, number one, these carbonates are multicrystalline. It's not one big crystalline. They're polycrystalline carbonates. And you can see the different textures there are different crystals in the carbonate. Number two, there's not much porosity in the carbonate. If we looked only at the top, we would see a lot of porosity. It looks like Swiss cheese. But when we cut it with this focused ion beam, it is fairly compact and not porous. And this gets into how it was formed. It had to be formed by crystals growing together and filling up the space. And again, the dark areas are the iron-rich magnetites. This shows the low porosity, even at the tens of nanometer scales. This shows that there's some sulfur-rich cross-cutting veins here. Again, we think that the original carbonate was shattered up and water flowed through it and deposited stuff. And that stuff includes sulfide minerals and some of the magnetites. The very interesting part of these magnetites is that they're embedded throughout all these parts of the carbonate that I've been showing you. That is, the iron-rich magnetites are found in the center and they're found on the rims. The difference is one of abundance. And that gets into how they got there, how they were formed. We would argue that they're not formed from the material that was already there. And that's what our critics have argued for several years, and a number of papers that they're formed directly from the material that was there originally, the carbonate. This shows that in the iron-rich rim, there are a lot of magnetites, which are all those little dark speckled things, and in the center, there are also magnetites. What are those magnetites doing there in the center? Remember, that center is mainly calcium carbonate. It is not iron carbonate. And so how do you get these magnetites from calcium carbonate? And we argue that you cannot do it. They had to be put there. And that is part of the key to our philosophy of how this all goes together. We've done a lot of work on the morphology of these little magnetites using some pretty advanced techniques, and I won't get into the details of it, but we're able to do crystallography on them, and these things are only like 50 nanometers, and we had to go up to Denver to one of the medical labs and use the tomography system connected to their electron microscope to get these images, and then they're reconstructed by computers. But we think we understand it fairly well, and we also argue that the magnetites produced by some of our critics are not like the ones that I just showed you. They do not have the elongation. They do not have there's no evidence for the proposed truncated hexaoctahedral geometry that we see in these Allen Hills magnetite. And this is some of their published data.
David, how similar are these morphologies to the magnetite crystals found not just in magnetotactic bacteria, but in pigeons? Aren't there a couple of organisms that have crystals like that on the Earth?
There are other organisms that have nanophase magnetite, and Kirschvink has found some in human brains and the theory is that magnetites in your brains, if properly connected to the brain cells, may give you a sense of direction that keeps your orientation. And certainly carrier pigeons have magnetites in their brains, and they use them. It's been demonstrated that they use them to track the magnetic field of the Earth, and that's part of they have other mechanisms. They have multiple mechanisms apparently for direction finding, but one of them is these little magnetites. And I think there are probably other animals which have not been documented or that I don't know about, which also have these magnetites in the brains. So the presence of these magnetites on a magnetic planet like the Earth and early Mars is really important, because it tells us that evolution had developed this mechanism for direction and position finding early on, even in very primitive things like bacteria. And furthermore, as Kirschvink argues, the most efficient magnets are not little cubes. They're somewhat elongated, like bar magnets. Now, magnetite itself is not normally elongated. It is only because of the envelope that bacteria produce and force the magnetite to grow within this envelope that bacteria can produce elongated bar magnets. And that is important because they're much more effective at reacting to the magnetic field. And we argue that the Martian ones have this elongation and the synthetic ones do not, or at least they've not been shown to. And we've written like 3 papers on that, and our critics have written three or four more papers, but we can I think show that our data are correct and our critics' data are at most problematical. The other important point, I don't want to talk about crystallography because that turns off everybody, including us, me. But the interesting thing has to do with the chemistry. And the chemistry of these is very critical. It turns out that the chemistry of these little magnetites is highly influenced by the chemistry of what forms them. There's a term used in biology, you are what you eat. Well, we have another term that you are what you heat. Because you can form magnetites out of carbonates by heating them, but the chemistry always reflects the starting composition of the carbonate. And we have done a lot of work on that, as have others, and we know that at different temperatures, different composition of carbonates will decompose to form magnetite. This shows some of those temperature graphs versus the composition, and you can see that at the very lowest temperatures, the iron-rich ones decompose, and the higher temperatures, the other ones decompose. If you decompose a carbonate with magnesium in it, you don't get pure magnetite, you get these magnesium ferrites. And again, you are what you heat. So you cannot get pure iron magnetite by decomposing an impure carbonate. And that is very critical to our argument, and we consider that fatal to the argument of our critics. I won't go any more into that. Well, this is a cartoon that demonstrates it pretty nicely. If you heat up on the left a mixed carbonate, what you get on the right is a mixed magnetite or spinels. Once you put a lot of other stuff in them, you don't call them magnetites anymore, you call them spinels. And that's what is illustrated there on the right with iron, magnesium, manganese in it. What you don't get is this on the bottom right, which is a pure iron magnetite. That's never been observed in anybody's experiment that we know of. And that's what our critics are trying to say is the cause of our magnetites. All right, I don't want to push this anymore, but this is critical to our argument. The origin of these magnetites is really vital to the whole question of life on Mars. This is for those of you who have had already thermodynamics. If you take the composition of carbonates and lower the free energy to the lowest energy state, and you can do that by heating it, for example, you get something like this in the lower left corner. The carbonates essentially are only happy when they're either the middle blue line there, which is essentially the dolomite line, half calcium, half iron, magnesium, or the lower right corner, which is iron plus manganese. Compare that, which is the low energy state, to the actual data on Allen Hills. The measured composition on Allen Hills is on the right. It is nothing like the low energy state. The one on the right is from Antarctica. It was found out by the Japanese expedition, and it probably lived in Antarctica for a few thousand years. We're not sure how long, maybe 10,000 years. And yet we see that the features are very similar. They're essentially, they're hollowed out places the size of typical microbes. In many cases, they're partially filled with remains of something, and these remains include some carbon-rich material and some iron oxide-rich material. And we would argue, and I'll show you some more pictures, that those are very similar to each other. And in fact, these meteorites have about the same, developed at the same time on Mars. We don't know where they came from, but they're both knocklights, and they came to Earth from the same area, possibly on Mars. And at least they were formed on Mars at about the same time. And so we would argue that if these are truly microfossils, they may represent similar consortia on Mars that formed these things a few 100 million years ago. And there's some elongated forms. Here's the Columbia River basalt. This is a biologic feature. And then we see a similar thing in Nakhla and Yamato 593. Again, seeing these does not prove they're microfossils or biology, but what it does is provide a tracer, a target for additional studies of these features and similar features, and we think that eventually we can determine if these are truly biologic or not. So it's important to assemble a lot of these tracers. These are interpreted to be Archean 3.4, 3.5 billion year old microfossils from Australia by Frances Westphal and her friends. And what we argue is that there are morphologically some very similar things in both Nakhla and Yamato 593, and Columbia River basalts. We're pretty certain that these are true biologic features on the left. A little bit of a biofilm there. We don't quite know whether that's indigenous or not. It's in Nakhla. And Yamato has indication of a biofilm as well. Again, these little bumps are found in both meteorites, and they're rather similar. Keep in mind that these are two totally different terrestrial environments. They're Antarctica on the ice and Egypt in the mud paddies collected within weeks, days, a few years. Just a few more comparisons here. These are quite common. As we look in these two meteorites, if we look carefully, we see these things all over the place. These are not just rare little things we happen upon. I can find these in almost any chip you give me of either one of these meteorites. Just some more comparisons. Storrs Lake in the Bahamas, these are recent microbial forms. On the left is some of the microbes in the carbonate sediments of Storrs Lake. On the left are recent ones. On the right are Yamato 593, the Mars meteorite from Antarctica. Storrs Lake and Yamato again. And we have chemistry on a lot of these. I didn't want to get into the chemistry, but we can show that there are interesting distinguishing features about these Yamato biomorphs that match the Storrs Lake ones. Back to Nakhla, showing how some of these features, which may be, which we call biomorphs now, I won't say that they're fossils, but the point is that these are not late contamination that just laid around here. These are built into the rock. They have become part of the rock. They have recrystallized. They are turned into oxides, and they're welded or embedded in the rock itself. And the rock, everyone agrees, is from Mars, and the people will accept that rock there is from Mars, but they won't accept that any of these features you see on it are from Mars. So I think there's a problem there. Nakhla, Again, some more Archean fossils, known fossils from Australia. Well, that's enough of those. We get really excited about these sometimes. There are some things that could be cyanobacteria. I want to make the point that these are two Martian meteorites having totally different terrestrial histories. Antarctica versus mud paddies of Egypt, and yet they contain incredibly similar features. Now, if these features are biogenic, it is very difficult for us to see how, under these two different environments, Antarctica and the mud paddies of Egypt, how these features could form to be identical in those environments. But we're not saying they're fossils at this point. But what we're saying is that these are pointers for more exhaustive analysis using chemistry, using TEM, using nanosims, which is an ion microprobe that works on nanometer-sized material. And incidentally, if you've seen the latest issue of Astrobiology journal, it has, not the very latest, but a couple journals back, it has some of our work out of our lab on these Australian microfossils, in which we were able to show that the Australian microfossils, which are known fossils, everybody agrees they're fossils, they contain carbon and nitrogen and phosphorus in their chemistry, and this material is associated only with these biomorphs, not with any part of the rest of the rock. And we argue that shows that those elements can be used to document that these are true microfossils. Now, in this case, we already know they're microfossils. But the next step, what we're going to do this summer, is look at some of these Mars meteorite features that I've shown you with the ion microprobe, and we'll look for things like the exact location of the carbon and the nitrogen. Nitrogen is a very unusual element because most rocks do not have nitrogen in them. Most minerals do not have nitrogen. The biggest source of nitrogen in rocks is life. And if you see nitrogen concentrated in these biomorphs, to us that is pretty certain indicator of life. So we're going to be looking this summer at some of those Mars features. Well, I want to finish up here and simply say that none of the original parts of our hypothesis have been discredited, and I will be happy to answer questions on any of these areas. The most controversial part that has been used to discredit us has been the presence of those magnetites. That's why I spent so much time on them, because that has simply been the strongest argument against us. And and we would argue that those magnetites, if you just find them on Earth, have been used for 30 years now as a certain biosignature. Once you characterize them and they have those properties, there are people like Joe Kirschvink at Caltech who will say that is a certain biosignature. That is a very good biosignature. And yet when we found them on Mars with those exact same properties, that construction dropped away, and particularly when it was suggested you could make those magnetites by this other non-biologic process. We have spent a lot of time, and we think we're correct in showing that the magnetites you make by these non-biologic processes are not like the ones in the Mars meteorite, and particularly when you try to make them from those mixed carbonates. That's what the rest of this says. And then at the end we say that other Martian meteorites contain biomorphs which are very similar and suggesting fossilized bacteria on Mars. That's it. That is the end. Thank you very much.
I know that was a lot of really technical stuff, but it was absolutely stunning. I mean, think for a moment. Mankind has wondered if he was alone in our solar system, in our galaxy or universe for centuries, millennia probably. And along comes Dr. McKay and says, we have some dead micro-Martians in our hands, or signs of it. What does it take to get everyone to believe that? And so you see it takes a lot of very careful chemistry and geology and knowledge of biology. And those are the arguments that he's trying to go through. I don't expect all of you to understand all of the geology and chemistry. But think, what would it take to try to prove this? Because extraordinary claims require extraordinary proof. So I'm going to leave this open if people want to ask questions.
You talked a lot about temperature formation at the beginning of your talk, and I was wondering, why is it that the heat of exit or the heat of re-entry wouldn't affect your biological
Because during entry into the Earth's atmosphere, that happens so fast, and the meteorite is so cold, having been in outer space, that only a very tiny skin gets heated, and that skin melts and forms a fusion crust. If you go in 5 millimeters to a centimeter, the meteorite is still cold in the interior. In fact, people have picked up fresh meteorites and gotten frostbite off of them because the center is way below zero. And the conductivity, it's just a kinetic thing. The conductivity does not keep up with the heating from the outside.
What is the consensus or speculation on what type of organism made these indentures? And then if, like, could you speculate if this life form might have evolved into something else by now or gone extinct?
All right, that's a real good question. And one of the arguments that is used by skeptics is that, hey, these magnetite-bearing microbes took a long time to evolve on Earth. What are they doing in this 3.9 billion year old, very old rock from Mars? And what would they do after that? I think there are two answers. One is somehow life may have started earlier on Mars, and this evolution may have taken place while Earth was still very inhospitable or even being sterilized by much more energetic impacts. And so Mars got a head start on us. That's one idea. Another idea is that the Mars life came from somewhere else. That's panspermia, that evolved somewhere else, and meteorites from who knows where came to Mars and Earth. And but then what happened to life if it got started that early on Mars? I think the answer is Mars is so inherently inhospitable to life that it really didn't have much room to evolve. It never had a big oxygen atmosphere that we can tell, although some people think it did early. It certainly hasn't for billions of years. The surface was always bathed in strong UV light. When the magnetic field died away at around 3.9 billion years, then radiation from the sun, particles could impinge on the surface of Mars and finish off any life that was there, perhaps. Mars was just not a friendly place at the surface. Now, we have always felt that life has continued underneath the surface on Mars, but it's hard. On Earth, we don't see any evolved forms underneath the surface. It's always just microbes. And so I think, in fact, on Earth, the really evolved forms did not appear until fairly late in the game. Certainly the multicellular forms that we're all familiar with are fairly late in the game, and on Mars there was just no time or space or environment that allowed those to develop. So I don't expect to find highly evolved things, although I would not be surprised to see some multicellular, a little bit more complex forms than anything I've shown you from the meteorites. Yeah.
I just wanted to make a comment.
Yeah.
So, I work with a geologist who is a skeptic.
Yeah.
And he thinks that he's found a very promising Earth analog for these carbonate globules.
Yeah.
And so he went to Svalbard, which is an island owned by Norway. It's very
Yeah.
high up in the Arctic Circle. And he found carbonate globules that look amazingly similar to
Yeah.
the ones that you guys found in ALH.
Yeah.
And it's sort of the project is just in its beginning stages, but I run the L2MS instrument now in Digzer's lab, and we find signatures from these inclusions that look almost identical to the ones in ALH, and also very much similar to the ones in another Martian meteorite, Dar El Ghani 476, with the exception that in the Earth analogs we find the low mass aromatics that are missing from the Mars meteorites. But I think you can explain the absence of those in other ways. I just wanted to make that comment.
The problem I've always had with those nodules from Svalbard is whether there was biology involved in their production too.
Yeah, I don't think they found any of the same biomorphs. I'm not aware of any, at least.
I think the absence of biomorphs does not mean that biology did not participate. I think there's got to be a more positive test of whether biology participated in their precipitation and growth.
Mm-hmm.
And I've heard a number of talks by that group, and I've asked a couple of the talks, well, what can you say about biology? Did biology participate or not? And normally they don't know.
Mm-hmm.
And I don't know if your data will help pin that down or not. But if it is very difficult using natural samples to show that biology has not participated. And my old geology professor said that biology participates in all low temperature chemical reactions on the Earth. And that's kind of the extreme view, but it shows how difficult it may be to exclude biology. And the only way you can really exclude biology is to do it in a sterile laboratory. So I don't know. I'm interested to see what new data comes out of that, and if they can somehow show that there's no biology, and yet you're getting this zoo of paws in there. That's great. Let's bring that data out and see what.
Yeah, I think he's planning soon.
Yeah, but you see my point that excluding biology is tough.
Any other questions?
Evidence from an asteroid has the advantage of pulling material from deeper in the surface of Mars. If you could take that ½ kilogram that's going to cost however many hundreds of billions of dollars in the future back from Mars, where would you take it from? If right now you could snatch a ½
In the light
kilogram.
of what you said, I believe that the deeper you can go, the better off you are if you're looking for life. If you're looking for how dust blows around and sand dunes form, then sure, get surface material. But your only chance of getting real life, I think, is to go down 1 meter, 2 meters, 10 meters. I would like to see, 100 meters. And on Earth, we're finding now in these gold mines, for example, that up to 3 kilometers, there's abundant microbial life going on in some of these subsurface environments. And why not on Mars, too?
Great question. Let's thank Dr. McKay.
For more, please visit us at stanford.edu.